Hi everyone, welcome to Bite Size Med, where we talk about quick, bite-sized concepts and basic medical sciences for study and rapid review. This video is on the oxygen-hemoglobin dissociation curve. The purpose of respiration is gas exchange. Oxygen has to go from the alveolus into the capillary, and carbon dioxide has to leave the capillary, get into the alveolus, and then get expired. Oxygen in the capillary gets transported in blood to reach the tissues which need to use it. This transport happens in two forms, as dissolved oxygen and as oxygen bound to hemoglobin. Now 98%, that's the bigger portion, is bound to hemoglobin. A bound gas does not exert a partial pressure, so this portion does not contribute to the partial pressure of oxygen. The dissolved gas, which is around 2% of the total oxygen content, can exert a partial pressure. The concentration of oxygen as a dissolved gas can be calculated by Henry's law, which is the concentration is equal to the partial pressure into the solubility coefficient. For oxygen, the partial pressure is 100 and the solubility is 0.003. So that's 0.3 ml of oxygen per 100 ml. And that's small, that's not enough for tissues. And that's why bound oxygen is more important for tissues. Hemoglobin has a heme moiety and 4 polypeptide chains, 2 alpha and 2 beta for adult hemoglobin. Each of these subunits can bind to one molecule of oxygen. Oxygen binds to the heme moiety. So that's 4 molecules of oxygen that can bind to one molecule of hemoglobin. Now, if all 4 are bound, that means it's 100% saturated. No more can bind. If it's just 2, it's 50% saturated. The binding of oxygen to hemoglobin is reversible. When it's bound to oxygen, it's called oxyhemoglobin, and without it, it's called deoxyhemoglobin. The oxygen-hemoglobin dissociation curve, which is a graph that shows the binding of oxygen with hemoglobin at different partial pressures. So along the horizontal axis, we have the partial pressure of oxygen, from 0 to 100. Along the vertical axis is the saturation of hemoglobin, again from 0 to 100. So the curve normally is sigmoidal. It's not a straight line, and that's because of something called positive cooperativity. The affinity of hemoglobin to oxygen changes with each molecule. Each molecule helps the next one bind easier. So hemoglobin is tetrameric, 4 molecules bind. The first molecule binds and increases the affinity for the second to bind. That increases the affinity for the third and then for the fourth. So there's positive cooperation. Versus myoglobin, which is monomeric, binds to a single molecule of oxygen. So there's no positive cooperativity, and therefore the curve is not sigmoidal. The P50 is the partial pressure of oxygen at which 50% of hemoglobin gets saturated. If the P50 increases, which means it moves to the right, there's lower affinity. More oxygen is needed to get to 50% saturation. If the P50 is lower, that's on the left, it increases affinity. So there's less oxygen needed to get to 50% saturation. In the alveolar air, oxygen has a partial pressure of 100. Oxygen diffuses from the alveoli into the pulmonary capillaries until it equilibrates at 100. This becomes systemic circulation. So the arterial partial pressure of oxygen is 100 millimeters of mercury. After tissue utilization of oxygen, the venous blood, which comes back, is at 40 millimeters of mercury. So in the lungs, where the oxygen is at 100, the hemoglobin is almost 100% saturated. So it's a high affinity. It's tightly bound. Because more oxygen is bound to hemoglobin, there's a reduction of the partial pressure in the pulmonary capillaries, because partial pressure is exerted by free dissolved gas, not from bound gas. So what this does is it keeps the oxygen partial pressure low in the capillaries while it stays high in the alveolar air, so it maintains a gradient, and oxygen continues diffusing, loading itself onto hemoglobin. In the tissues, the partial pressure of oxygen is low. It's at 40 millimeters of mercury. So on the graph, we can see that hemoglobin is 75% saturated. It's got a low affinity. Oxygen is loosely bound and can be unloaded easily onto tissues. Lower affinity, easier release of oxygen. The oxygen dissociation curve can shift to the right or to the left. Shifting to the right happens when there's low affinity. There's more release or unloading of oxygen. So right releases, R for R. What happens to the P50? That increases because 50% saturation is achieved at a much higher partial pressure of oxygen. So when does the curve shift to the right? First, when there's high partial pressure of carbon dioxide and a low pH. Tissue metabolism produces more carbon dioxide, so it increases the PCO2. That increases hydrogen ions and lowers the pH. Carbon dioxide binds to a different site on hemoglobin than oxygen, but it lowers the affinity for oxygen. So there's more oxygen release and delivery to the tissues to meet the oxygen demand from excess tissue metabolism. This is the Bohr effect. It's that binding of carbon dioxide to hemoglobin reduces the affinity for oxygen and shifts the curve to the right. In exercising muscles, there's increased lactic acid production, which lowers the pH. There's also an increase in temperature. Both of these also shift the curve to the right, so more oxygen delivery for higher oxygen demand of these exercising muscles. A byproduct of RBC glycolysis is 2,3-diphosphoglycerate. This binds to the beta chains of deoxyhemoglobin, reducing the affinity for oxygen. So high 2,3-DPG can shift the curve to the right. In hypoxia, like in high altitude, there's increased 2,3-DPG production, which reduces the affinity for oxygen, increasing release, and the tissues need more oxygen, so it meets the demand. So high PCO2, low pH, high temperature, high 2,3-DPG, hypoxia, and high altitude can all shift the curve to the right. So the opposite will shift the curve to the left, where the affinity is high, the release is low or the unloading is low, and the P50 is also low. That's in low PCO2, high pH, low temperature, low 2,3-DPG. Another thing that can shift the curve to the left is in fetal hemoglobin. Adult hemoglobin has 2 alpha and 2 beta chains. Fetal hemoglobin has 2 alpha and 2 gamma chains. 2,3-DPG binds to the beta chains, does not bind avidly to the gamma chains, and so there's reduced binding of that, increasing the affinity for oxygen. So fetal hemoglobin has a higher affinity for oxygen than adult hemoglobin. If this video helped you, give it a thumbs up, share, and subscribe. Thanks for watching, and I'll see you in the next one.